Hello, everyone, and welcome to today's webinar, Current Landscape and Future Directions of Multi-Specific Antibodies. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sino Biological. To learn more, visit www.sinobiological.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Shebna Massey, Associate Product Manager, Sino Biological. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Massey, you may now begin your presentation.
Thank you for the introduction. Hi, everyone. I am Shebna Massey. I am Associate Product Manager at Sino Biological. Today, I'm going to talk about the current landscape and future directions of multispecific antibodies. To cover this topic, I will give a brief overview of multispecific antibody therapy. Then we'll talk about the FDA-approved bispecific antibodies, followed by a discussion on new advancements in antibody-based therapeutics and what multispecific antibodies are in various stages of clinical trials. And after that, we'll discuss the challenges and future directions of the multispecific antibody therapy. And finally, I will introduce how Sino Biological is supporting the research in antibody therapy development. So let's begin with the overview of multispecific antibody therapy. Antibody therapeutics is a growing market. Therapeutic antibodies have become the predominant class of new drugs developed in recent years. It has been successfully growing since its introduction more than 4 decades ago. And over the past 5 years, antibodies have become the best-selling drugs in the pharmaceutical market. The global therapeutic monoclonal antibody market was valued at approximately $115 billion in 2018, and it is projected to reach more than $300 billion by 2025. Thus, the market for therapeutic antibody drugs has experienced explosive growth as new drugs have been approved for treating various human diseases, including many cancers, autoimmune, metabolic, and infectious diseases. With this growing market potential of antibody therapy, the antibody engineering has dramatically evolved as well. At first, whole naked IgG antibodies were used directly as therapeutics. Now, therapeutic antibodies can be roughly separated into 2 categories. The first category involves the direct use of the naked antibody for disease therapy. These antibodies are used for cancer treatment and elicit death by different mechanisms, including ADCC or CDC. The example for that is the drug Rituxan, and or the direct targeting of cancer cells to induce apoptosis. The example for that is Herceptin, or the targeting of the tumor microenvironment, or targeting immune checkpoints. And so this is one way, the one category of antibody therapeutics. The second category for antibody therapeutics now includes additional engineering, where this additional engineering is performed to enhance their therapeutic efficacy. Some general approaches for the use of these antibodies includes immunocytokines, antibody-drug conjugates, radionuclides, immunoliposomes, bispecific antibodies, and CAR T cells. An example for bispecific antibody is Blincyto, and for CAR T cell, Kymriah is a popular example. Our focus for this talk will be bispecific and multispecific antibodies. So multifactorial diseases have surged the need to enhance the potency of monoclonal antibodies to further extend their mechanism of action. Multispecific antibodies are emerging enhanced counterparts of monoclonal antibodies which have the ability to specifically bind with 2 or more different antigens. And by doing so, they increase their selectivity towards the target cell and also lead to enhanced cytotoxicity. This schematic here shows the different multispecific antibodies binding to the antigen on the tumor cell compared to the classical monoclonal antibody. The bispecific and trispecific antibodies here can bind to the antigen on tumor cells, as well as they can recruit T cells and other immune cells at the tumor site, and that increases its specificity, clinical efficacy, and potency. There have been important advances in antibody engineering over the past decade, and that has enhanced the safety and efficacy of therapeutic antibodies. These developments, along with a greater understanding of antibodies, have paved the way for next generation of new and improved antibody-based drugs. There are various formats of bispecific and multispecific antibodies, as you can see here. There are combinations of light and heavy chain variable fragments, as shown here in panel 1. You can see different bispecific and multispecific antibodies. They are in the IgG-like format. Whereas in panel 2, you see that there are bispecific antibodies that are fragments and fusion proteins. So overall, there are various formats of IgG-like and fragment multispecific antibodies. What are the advantages of multispecific antibodies over traditional monoclonal antibodies? So multispecific antibodies have many advantages. They offer enhanced specificity towards the target, and they have, as well as they have better therapeutic effects. Their ability to engage with the immune cells has enhanced cytotoxic effects, and they can redirect specific polyclonal immune cells such as T cells and NK cells to tumor cells to enhance tumor killing. And simultaneously, they are better at preventing off-site toxicity, given their specificity towards the target antigens. And from the perspective of healthcare costs, multispecific antibodies are appealing since a similar or superior therapeutic effect could be achieved with a single therapeutic agent in case of multispecific antibodies, as compared with using a combination of monoclonal antibodies. So this effectively reduces cost in terms of development and production when compared to multiple single-based antibodies used in combination therapy, or even compared to the production of CAR T cells, which currently require a several weeks long process for each individual patient. So multispecific antibodies they have enhanced tumor killing, they have enhanced specificity. Also, their dual targeting for immune checkpoints, it is unique and it has overlapping functions and effectively prevents drug resistance. And compared to other treatments, antibody-based treatments, multispecific antibodies are also cost-effective. Now, majority of the antibody therapy efforts have been targeted towards cancer therapies. So let's take a quick look at the landscape of multispecific antibodies in cancers. And first, we'll begin with the hematological cancers, hematological malignancies. So in our previous schematic, we saw that multispecific antibodies recruit immune cells in addition to binding the tumor antigens. So they are being developed as these multispecific antibodies in hematological malignancy. They are being developed as immune cell engaging antibodies, and they utilize CD3. So that is the major antigen binding that the multispecific antibodies in hematological malignancies have. That is CD3. And as you can see here, that this figure shows that 129 out of the 138 bispecific antibodies from the past few years have been directed to targeting CD3. This direct engagement of T cells helps to reduce the time and gap between an immune cell and cancer cells. A few others from the past few years have also bridged the gap between NK cells to the tumor cells, and by targeting the CD30 and CD16A. The other most promising targets for multispecific antibodies are CD19, CD20, and BCMA in hematological malignancies. So there are different combinations of these multispecific antibodies that have been tested and shown to have efficacious effect in clinical testing, in preclinical and clinical testing. All right, so on the other hand, in solid tumors, what does the landscape look like for multispecific antibodies? So overall, solid tumors account for 90% of the newly diagnosed cancer cases. And currently, there are very few drugs available to produce a durable therapeutic benefit in patients. And that is because of the high heterogeneity of cancer cells. In the past few years, there have been more than 170 clinical trials of bispecific antibodies to treat solid tumors. And in these 170 clinical trials, the popular targets in solid tumors have been, we can see here, CTLA-4, PD-1, PD-L1, HER2, EGFR. These are some of the popular targets in solid tumors. So overall, this also shows that there are different combinations for bispecific antibodies that have been used in terms of targeting the antigen targeting combinations that have been focused on in clinical trials for solid tumors. Now, following this overview of multispecific antibodies in hematological malignancies in solid tumors, let's look into what multispecific antibodies have been approved by the FDA. So here we can see that currently there are 9 bispecific antibodies that have been approved by the FDA. We can note some popular target molecules like CD3, CD19, EGFR, MET, VEGF, BCMA. These are some popular targets that we can see in this list of these FDA-approved bispecific antibodies. And we can note that there are, in terms of the indications, majority of them are for hematological malignancies. And there are also 2 of these drugs that the multispecific antibodies are for non-cancer indications. One is for hemophilia A, and the other one for macular degeneration. And all of these have been approved between the years 2014 and 2023. And I think it's worth noting that just this year in 2023, there have been 2 bispecific antibodies that have achieved approval by the FDA. Now that we have seen what has been approved by the FDA, let's take a look at what multispecific antibodies are in different stages of drug discovery and clinical trials. So in this table, you can see that there are, so it's showing 9 out of the 15 bispecific antibodies that are in the late clinical stage of clinical trials. Some target antigens that we can note here are the popular HER2, PD-L1, CTLA-4, VEGF, EGFR, BCMA, CD3, but also some new targets such as DLL4 and FcγR receptor 2b are also worth noting here. And another thing that is worth noting here is that there are several bispecific antibodies in these late clinical trial stages that are targeting for solid tumors. So that is a very promising advancement in multispecific antibodies, to see that there are more of these bispecific antibodies that are in the pipeline for targeting solid tumors. So overall, we know that there are over 50 bispecific antibodies that are under investigations in clinical trials, and more than 180 bispecific antibodies that are in preclinical development. Now, specifically looking at the developments of trispecific antibodies, there are 7 trispecific antibodies that are in early clinical trials. And of these 7, almost half are for hematological malignancies and half for solid tumors. And these trispecific antibodies, they simultaneously target 2 tumor-associated antigens, and in addition, they co-engage T cells by binding at the CD3, and along with agonists of co-stimulatory molecules or antagonists of co-inhibitory immune checkpoint receptors in a single molecule. So this may revert T cell exhaustion that is very commonly seen in antibody therapies. And similarly, targeting of 2 activating receptors in NK cells may improve their cytotoxic potency. So different combinations of these targeting antigens can help further tune the efficacy and potency of these multispecific antibodies. So even though there are no trispecific antibodies that have been approved by the regulatory bodies, we see that there are several of these antibodies that have entered the initial stages of clinical development. Now, looking at the exciting advancements in bispecific and trispecific antibodies, it is even more exciting to see that there are also tetra-specific antibodies in preclinical developments. And as the name indicates, that these antibodies can bind 4 antigens simultaneously. And there are 3 tetra-specific antibodies in preclinical research, where they have the ability to provide superior potency by binding 4 different receptors simultaneously. They also have more challenges to overcome as well. And a few of these challenges include overcoming the manufacturing instability. So these 3 tetra-specific antibodies that are in preclinical studies, the applications for those, so they are majorly for cancers, leukemia, glioblastoma, lymphoma, and solid tumors. So it is very exciting to see that the multispecific antibodies are making such exciting advancements, and tetra-specific antibodies are also in preclinical development. We have briefly touched on how tetra-specific antibodies are facing challenges. Now, let's see what kind of challenges are multispecific antibodies overall facing, and also the future directions for the multispecific antibody therapy. So despite the clinical success of bispecific antibodies, many obstacles remain, such as dosing, treatment resistance, and modest efficacy, especially in terms of solid tumors, which is mostly because of extracellular scarcity. So all of these challenges still need to be overcome. And even though relative cost, they're relatively cost effective, we have previously seen and talked about that in the advantages of multispecific antibodies, it is still a very long and costly manufacturing process that needs to be further optimized. And the aggressive stimulation in multispecific antibody therapy it has the risk of eliciting the cytokine release syndrome. So that is something that still needs to be further addressed for multispecific antibody therapy. And as for many therapeutics, it is always challenging to determine the best route and optimum dose for administration, as well as addressing the systemic side effects and the even controlled release of formulations. So all of these things are some main challenges that need to be overcome for the advancement of multispecific antibody therapy. Some examples of challenges seen in multispecific antibody therapy for solid tumors is that, as seen in other immunotherapies, there is increased on-target off-tumor toxicities. There is sparse T-cell infiltration and impaired T-cell quality due to the presence of an immunosuppressive tumor microenvironment, which affect the safety and limit efficacy of CD3 multispecific antibodies. So some ways to overcome these hurdles is by targeting the tumor-exclusive antigens, such as human leukocyte antigen, HLA, presented neoantigens, or surface antigens from virally induced cancers. The number of increase of intratumoral T cells and the improvement in the quality of can improve the quality of T cell responses. And one way to achieve that is by blocking the immunosuppressive cytokines and targeting co-stimulation of T cell effector function in survival. So these are the few ways that are being utilized to overcome this, different hurdles in seen in multispecific antibody therapy. What does the future of multispecific antibody look like? The commercial clinical pipeline of antibody therapeutics has shown substantial growth over the past years, as we have seen by seeing different bispecific antibodies that have already gained the FDA approval, and then multiple that are in different stages of clinical development. So despite all the challenges, the future of multispecific antibodies is very promising. Some future directions under focus are enhancement of safety and tolerability, potential of the combinatorial approaches that can help to overcome the challenges, and it can generate more effective responses. Also establishing various antibody platforms, including Fc engineering, different combinations of heavy and light chain pairings, and the high throughput production methods that can help improve the cost effectiveness of in multispecific antibody therapies. And also extended it. So we have seen that the majority of them have been focused on cancers, hematological, and solid tumors. So in the future, it is being focused on taking it beyond oncology and extended it to different disorders and diseases, neurological, infectious, ophthalmological. We saw that one indication included ophthalmology, but it is focused to extend it to beyond oncology more, and also cover rare diseases and etc. All right, so now I would like to take the opportunity to highlight how Sino Biological, as a leading reagents company, is supporting the multispecific antibody therapy research. I would like to begin with sharing that Sino Biological is a one-stop supplier of proteins, recombinant proteins, antibodies, gene products, and ELISA kits, and more. We specialize in recombinant proteins and antibodies. Within the recombinant proteins, we offer drug target proteins, GMP-grade cytokines, CAR T cell therapy targets, immune checkpoints, and Fc receptors. These are a few major product lines that I would like to highlight. And then for our antibodies, we offer primary and secondary antibodies, tagged, untagged versions of antibodies, also loading controls, and neutralizing antibodies. We are also very proud to offer the world's largest viral antigen bank. We offer 2,000 unique antigen and antibody products. In addition to our catalog collection, Sino Biological also offers customized CRO services for protein and antibody production and antibody development, including nanobody production, antibody humanization, AI-powered antibody affinity maturation, and others. So specifically for multi-specific antibodies, Sino Biological is equipped with the highly efficient and excellent platforms for antibody development, humanization of antibody humanization and affinity maturation platforms. And we have a very experienced team and offer a well-established antibody drug evaluation system. So Sino Biological provides a wide range of reagents and services for the entire process of multispecific antibody development. And I will go into more details of this in my next few slides. So as I said, we offer a wide range of products and services for customers with the complete multispecific antibody development solutions. And it covers the various phases of the development process, beginning from antibody development to antibody optimization, druggability assessment, animal model evaluation, and process development in clinical studies. Here, I would like to highlight the some target proteins, which are, target proteins, as we have seen, are key reagents for antibody drug development. So some examples of recombinant target proteins that Sino offers are PD-1, PD-L1, CD3, CTLA4, VEGF, EGFR, to highlight a few. And we offer different target proteins. This is a non-exhaustive list, just a few examples. We cover target proteins, immune checkpoints, cytokines, and receptors for drug development. And here are a few examples through which I would like to highlight that our recombinant proteins are high purity, and they are validated for their bioactivity, as shown here for two examples of CD3 and PD-1 recombinant proteins that are sec-HPLC. Their purity is determined by sec-HPLC, and they are validated for their bioactivity in binding assays. In addition to high purity and bioactivity validation, these recombinant proteins are also low in endotoxin and have high stability, and they are available in various forms, tag-free and available in options of multiple tags and from multiple species. And these recombinant proteins show high batch-to-batch consistency. And lastly, I would like to highlight that our products are very trusted by our users, and it is indicated by the high citation number for our products in top-tier journals. Sino Biological offers this recently optimized product line of Fc receptors, as we saw that Fc receptors are being targeted by multispecific antibodies, and they play a significant role in the various steps of antibody therapy research, with applications at the antibody screening step, candidate development, ADE, antibody-dependent enhancement effect, and they are applicable in various indications, the main one being cancer immunotherapy, and they're also applicable in vaccine development and immunizations. So as highlighted for our recombinant proteins, this product line for Fc receptors is also high in purity and validated for its activity in various tests and highly cited. Another important product line that is very applicable in multispecific antibody therapy research is cytokines and growth factors. Sino Biological offers premium quality cytokines and growth factors, and these are widely used in cell proliferation inhibition detection, cytokine release detection, mixed lymphocyte reaction in other experiments in multispecific antibody therapy development. And Sino Biological has developed a series of cytokines with high activity, high purity, and high batch-to-batch consistency, as indicated here through the example of IL-18 here. And this table shows some selected cytokines, cytokine catalog products that are well cited. And I would also like to highlight that Sino Biological products are high purity. These high quality products are very competitively priced. And just one example here that we show high batch-to-batch consistency, and our product is very competitively priced compared to other competitors. We also have developed a series of high-quality ELISA kits and ELISA pair sets that we offer. They are raised against full-length proteins, these antibodies, and it contains the capture and detection antibodies. Both are monoclonal, and these kits and antibody ELISA pair sets go through stringent quality control measures, and again, they are very affordably priced. In addition to our large catalog collection, we offer various custom services for antibody development. We have 4 antibody development platforms: phage display, Beacon single B-cell screening platform, FACS single B-cell sorting platform, and hyperdoma development platform. We also offer protein expression services. These are high throughput and large scale protein production services. And we offer 4 major expression systems and use the high efficiency expression vectors. In addition to that, we also have control antibody production service, where we use research grade antibody drugs, and they can be used as positive controls. And we have a track record of successful production of more than 10,000 antibodies. Then additionally, we also offer the stable cell line development service. We offer the officially licensed CHO K1 cell lines to support IND, NDA, and BLA applications. And this has a fast delivery within 2 to 4 months. And there are a variety of packages that are available. So overall, Sino Biological has an extensive catalog where we offer more than 6,500 proteins, 14,000 antibodies, more than 47,000 gene products, and more than 600 ELISA kits. And from recombinant proteins to monoclonal antibodies, our high-quality products undergo rigorous testing and validation to ensure reliability and consistency. And with our extensive portfolio, we aim to provide researchers with the necessary tools to advance scientific discovery and drive innovation in the field. Also, our CRO services are designed to support your research and development needs across various fields. We offer a range of services, including recombinant protein expression, as I've shared, recombinant antibody production, antibody development, and with our expertise and state-of-the-art facilities that we offer, we provide reliable and efficient solutions to accelerate your research projects. And with that, I would like to thank you all for your attention, and I'll be happy to take any questions. Thank you.
Thank you, Dr. Massey, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question is, what is the current status of multi-specific antibodies in terms of drug discovery and approval?
Thank you for the question. So there has been a remarkable increase in the number of antibody therapeutics in the past few years, as we have seen throughout this presentation. And currently, there are 11 bispecific antibodies in the late stages of clinical trials, so phase 2 and 3, as of July 2023. And the most recent approvals that have been are for a T-cell engaging trispecific antibody that targets the BCMA, CD38, and CD3. And it has been granted an orphan drug designation by the FDA, and it is for the treatment of multiple myeloma. So that with that, we have seen throughout the presentation how through the FDA and the late stages of clinical trials, and even in the early preclinical stages. So throughout this process, the multispecific antibodies are very rapidly moving through this. And just, within this year, 2 bispecific antibodies have achieved FDA approval.
Fantastic, thank you. Our next question that we have here is, what is the status of multi-specific antibodies and monoclonal antibodies in terms of commercial pipeline growth?
For the commercial pipeline growth of antibody therapeutics overall, it has shown a substantial growth over the past year. So comparing the antibody therapeutics development in 2022 and compared to that in 2023, there has been a 20% increase in the commercial pipeline, clinical pipeline for antibody therapeutics. And the overall number of the antibody therapeutics has dramatically increased as well. There have been more than 140 antibodies in the commercial pipeline just this year. And if we characterize them based on the indications, then definitely the majority of the antibodies have been for cancer treatment, as we noted during the presentation as well. But for the non-cancer indications, there is one bispecific antibody that is in the late clinical trials, and that is for the cardiovascular indication. And bispecific antibodies overall are very successfully navigating the late-stage clinical studies and the regulatory review. And as I just mentioned that, out of the 9 FDA-approved bispecific antibodies overall, 2 of those have gained the FDA approval just here. So the antibody therapy is very rapidly growing and moving through the commercial pipeline, I would say.
Okay, great. Thank you. Our next question here is, how does the selectivity of multi-specific antibodies towards multiple antigens enhance their cytotoxic activity and potential as cancer therapies?
The enhancement of cytotoxic activities and potential as cancer therapies for, by targeting multiple antigens for multispecific antibodies. So while multispecific antibodies are a very dynamic and promising field, it still faces some major challenges, as we noted. And the most serious difficulty is certainly the availability of the suitable target antigen for the majority of cancers. The common target in various bispecific and multispecific antibodies is CD3. And that allows for the direct recruitment of T cells to the tumor, and that enhances the cytotoxic effect. And that has been critical in showing efficacious responses in antibody therapeutics. CD3 has been a most promising target for multispecific antibodies in both hematological and solid tumors. So it is showing, really good cytotoxic activity and potential as in cancer therapies.
Wonderful, thank you. Our next question here is, does Sino Biological develop multi-specific antibodies?
Sino Biological offers a very rapid and efficient bispecific antibody expression services We have a strong expertise and experience in recombinant antibody production. We can initiate the production from antibody sequences and facilitate the expression of a variety of bispecific antibody forms, such as the bispecific T-cell engagers, the BiTEs, diabodies, cross-monoclonal antibodies, and DVD-IgG. Sino Biological provides antibody expression services ranging from microgram to gram level productions, and we use efficient expression vectors and optimized proprietary medium, transfection reagents, and high-density cell suspension culture technology to meet the diverse application needs of our customers. So we have this entire process through which we facilitate the development of bispecific and multispecific antibodies.
Okay, excellent. Thank you. And it looks like we have time for just one more question today. What services does Sino Biological offer for multi-specific antibodies clinical studies?
So all the, I mentioned these services during my presentation. Sino Biological provides services along the entire process of multispecific antibody development and characterization. And to support the process of development and clinical research of multispecific antibodies, we provide a comprehensive range of products and services. And this includes the cell line development service, the PK antibody development service, ADA antibody development service, and CHO, the CHO cell culture media. Starting from the customer's cDNA or antibody sequence, we can provide cell line development service that ranges from expression vector construction to screening for high yield and stable cell lines. And we offer the officially licensed CHO-K1 cell lines to support the IND, NDA, and BLA applications. And to further expand on that, we offer a range of anti-idiotype antibody generation service packages for the quantitative testing of drug levels in animal or human serum. And this facilitates the preclinical and clinical PK studies of multispecific drugs. And for this, we have 4 antibody development platforms, the hybridoma, phage display, Beacon single B cell, and FACS single B cell platforms. So overall, our services also provide the to perform the immunogenicity assessment of multispecific drugs in preclinical and clinical studies, and we do this by testing with anti-drug antibodies, the ADAs. And Sino Biological develops the high affinity, high specificity, and high sensitivity ADA antibodies for customers using the well-established rabbit polyclonal antibody technology that we have. So we cover, like, as you must be able to guess, that we cover the various steps of the preclinical and clinical studies for the multispecific antibodies.
Okay, wonderful. Thank you. Thank you, Dr. Massey, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Sino Biological, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.